All right, folks, so welcome to Chapter 1, our first chapter of microbiology, and this chapter is titled The Microbial World in You. So this, just like any first chapter in a textbook, is really just an overview of microbiology and what we're going to discuss kind of through the rest of the semester. So we're going to touch on a lot of broad concepts and introduce you to the concept of microbiology. Now, one thing that I like to put in here, some interactive slides like Q&A. Now, normally when this is held in an actual classroom, I can kind of get feedback, so I can't really do that because this is a recorded lecture. But I will present this Q&A and give you kind of a second to think about it, and then we'll have a sort of discussion. So advertisements tell you that bacteria and viruses are all over your home and that you need to buy antibacterial cleaning products. So the question is, should you? All right, so I'll give you a second. You can pause the lecture, kind of think about that for a second. Now, really, there's no real clear answer to this. I do want to point out some statistics. Seasonal allergies in the United States increased by 31% from the years 1985 to 1995. A couple other statistics. Childhood peanut allergies doubled from the period of 1997 to 2002. And lastly, asthma increased from a rate of 35 to 56 cases per 1,000 Americans, and that was during the year 1982 to 1994. So those are some big increases in allergies and asthma. What does that have to do with this Q&A, this slide? Well, one theory on this is called the hygiene hypothesis. And that is that as a very clean society, as an industrialized modern society, we're very clean. We use antibacterial products. We're very conscious of dirt and those sorts of things. And that may actually have a negative impact on our immune systems. So basically, the hygiene hypothesis states that normally throughout our evolutionary history, we've been exposed to a lot of microorganisms very early on in life, and that trains our immune system to recognize what is harmless and what is possibly disease-causing. And the fact that we're not being exposed to a lot of those early childhood organisms anymore may have a negative impact on our immune system. All right, so that's just, again, one hypothesis. Of course, the other side of the coin is we also have a lot less disease, infectious disease, than we used to because of things like cleaning products and being more hygienic in general. So there really, again, is no clear-cut answer to this, but I just want everyone to think about that as we go forward. That's really a theme that I want to bring up a lot in this class, is that microorganisms, bacteria, get a bad reputation. They're not all bad. In fact, 99% of microorganisms are not pathogenic to humans, and a lot of them actually, we're now realizing, have positive effects on human health. So we don't always want to think about microbiology as the study of infectious diseases. It's really the study of all microorganisms, and including a lot of the good ones. So anyway, let's start off with some terminology. Microorganisms, or microbes, is the term that we prefer to use when talking about organisms that are too small to be seen with the unaided eye. You may have heard the term germ. A lot of people use the term germ. Germ is not really a scientific word. It refers to a rapidly growing cell. So in this class, we'll try to use the words microorganism and microbe instead of germ. So what we're going to look at next is sort of an overview of microbes in our lives. What do they do for us? We know, of course, that a few are pathogenic, which means disease-causing. We're going to use that term a lot, pathogenic, so it's a good idea to make sure you know that. Other than that, though, like I said, the vast majority of microorganisms are not pathogenic. All right. Now, there's a whole lot of species of bacteria. If I asked you how many different species of bacteria are there, think for a second what you would say. All right. Now, the answer to that, we don't exactly know, of course, because we haven't discovered all of them, but there's an estimate that ranges anywhere from 10 million to 1 billion different species. And again, the vast majority are non-pathogenic, and many are actually beneficial to us. And if we look at the total number of individual bacteria on Earth, the total number of individual cells, it's estimated at 10 to the 30th power. All right, so a 10 or 1 with 30 zeros after it, that's about how many microorganisms there are, how many cells there are on Earth. So anyway, that just gives us an idea of the vast array of these microorganisms and where they are and what they do. Now, in terms of other than some being pathogenic, again, the vast minority, what are the other things they do? Some of them decompose organic waste. They're very important in recycling nitrogen and phosphorus and carbon in the environment. And they turn that nitrogen, phosphorus, and carbon into forms that plants and animals can use. So they're very important in that idea. Many of them are producers in the ecosystem by photosynthesis. So when you think of photosynthesis and what produces oxygen, you probably think plants. But the vast majority of oxygen made on Earth is actually made by algae and cyanobacteria, which are microorganisms. A lot of them we can use to do different things and produce different things that we like or use, like industrial chemicals, ethanol and acetone. They can be produced by fermentation of certain microbes. They can also be used to produce fermented foods like vinegar, cheese, and bread. So this is something that is centuries old, the idea of fermenting foods to make them last longer, to make them taste better, or even to get us drunk, right? Those are all fermented foods. And all of those are produced by microbes. They can produce products used in manufacturing and treatment. So we're using microorganisms nowadays to produce a lot of different medicines. Insulin, for example, is produced in E. coli. Human insulin is produced in E. coli. And in fact, it's not so far-fetched to say that we could actually have genes, all right, designer genes, Geans, not Genes, made by microbes. All right. In fact, right now, when you buy any kind of faded denim product, or sometimes they used to call it stonewashed denim, that used to be done by washing with stones. Now we use enzymes from a fungus called trichoderma. They produce these enzymes called cellulases, and cellulases digest cellulose, which is present in cotton, and it helps to fade them and to soften them. Now, these other ones are in the research phase. In theory, there's a type of bacteria called gluconacidobacter that can make cellulose, all right, which is really the structural component of cotton. A mushroom peroxidase, it's an enzyme that certain mushrooms produce that could actually be used to bleach fabric. In fact, indigo, the dye that's used to make blue jeans blue, can actually be produced in E. coli. That's what we're seeing here. These are genetically engineered E. coli that have been engineered to produce the dye indigo. You can see they're nice and blue. And in fact, they're exploring one substance called bacterial polyhydroxyalkanoate, or PHA, which is a natural substance that resembles plastic, but it's produced by bacteria. So even the plastic that makes up the buttons on our jeans or that packages the jeans could even be made using microorganisms. So I want to ask you folks a question. How else do we use microbes in our everyday lives? All right. This is an open-ended question. I'm not going to answer it, but I want you to think of that. Is what else do we use microorganisms for? We've kind of covered some of the basics, though. So it's important to have knowledge about microorganisms. That's kind of the whole point of this course, is to learn about microbes. Because in the past, if we look back at history, knowledge of microorganisms has allowed humans to do things like prevent food spoilage or prevent disease occurrence. All right, now these are some of the things that bacteria do that are bad, or microbes do that are bad. Again, there's plenty that we know that microbes do that are good, but that's some history there, is we didn't know what spoiled food for a long time. And if we know how it happens, we can know how to prevent it. It's also led to aseptic techniques that prevent contamination in medicine and in microbiology laboratories. Those aseptic techniques that you use in surgery and so forth and in the microbiology lab are very important, and it requires knowledge of microorganisms. We're briefly going to mention naming and classifying microorganisms, but we're going to talk more about that in Chapter 10. So it was a gentleman by the name of Carolus Linnaeus that established the system of scientific nomenclature, which is scientific naming. That's what that means. Now, you learned about this in general bio, but we know that each organism is given two names, the genus and the specific epithet. Scientific names are italicized or underlined. The genus is always capitalized, and the specific epithet is in lowercase. Scientific names are always Latinized. So even though no one speaks Latin anymore, any new scientific name, when we come up with one for a newly discovered organism is made to sound like it's Latin. All right, and we'll look at that in a second. That's on purpose. And scientific names are not random. They sound really complex, but they are either descriptive or they honor a scientist in some way. So they describe the organism or maybe even honor the scientist that discovered that organism. And here's a great example, E. coli bacteria, which is the abbreviated name for Escherichia coli. Escherichia coli, very common organism that lives in the human large intestine. That name is not random. It honors the discoverer, Theodore Escherich, so that's Escherichia, and it describes the bacterium's habitat, which is the colon, so that's the coli part. But you'll notice that we don't call the organism Escherichia colon, we call it Escherichia coli. So again, that's the whole Latinized thing. These names are made to sound like they originated in the Latin language. It's just tradition. Another example, Staphylococcus aureus, is an organism that causes what you would call a staph infection. All right, Staphylococcus aureus, a common human pathogen. It's descriptive, that name. Staphylo means clustered, and cocci means spherical. So Staphylococci describes how they look under the microscope. They are round cells, spherical cells, that are clustered together. And aureus means gold in Latin. All right. If you've taken chemistry, you know the chemical symbol for gold is Au. That's Latin because it's aureus is gold. So after the first use, scientific names can be abbreviated with the first letter of the genus and then the specific epithet. So, for example, Escherichia coli and Staphylococcus aureus are found in the human body. E. coli is found on the large intestine, and S. aureus is on the skin. So you can see that as long as we use the full name once, we can always abbreviate. And this is always a good thing to keep in mind whenever you're doing any writing in this class. So especially like we'll write lab reports, keep this in mind. You always have to use the full name once, but you can abbreviate thereafter. So microorganisms come in lots of shapes and sizes. In fact, we have lots of different types of microorganisms. We have bacteria, archaea, fungi, protozoa, algae, viruses, and multicellular animal parasites. And we're going to go through all of them. All right, starting with some pictures, which we'll come back to again. We always like to put pictures in here. But before we talk about the different types of microorganisms, I just want to remind everyone of what you learned about the two types of cells in general biology. All right, we know that any cell on Earth is either a prokaryotic cell or a eukaryotic cell. All right, so this is something that you learned in general bio, but prokaryotic cells like bacteria are smaller, they're very simple in structure, they have no membrane-bound organelles. They have DNA, but it's not in a nucleus, it's in a nucleoid region, which is not separated by a membrane from the rest of the cell. Whereas a eukaryotic cell, like our cells, are larger, more complex. They have many types of membrane-enclosed organelles, and they have, most importantly, a nucleus that surrounds the DNA. So let's start by talking about the different types of microorganisms. First with bacteria. We're going to focus a lot on bacteria in this course because bacteria are probably the most broadly understood of all the microorganisms. Bacteria are prokaryotes, and again, if you recall, prokaryote literally means pre-nucleus. They have no nucleus, no membrane-bound organelles. They have a cell wall that's made out of a substance called peptidoglycan, which we'll talk about in Chapter 10 more. That's an important characteristic of bacteria that helps to characterize them. They have peptidoglycan cell walls. They're single-celled. They replicate by a process called binary fission, which is much simpler than the process that our cells use to reproduce, which, if you recall, is called mitosis. For energy, it really depends. Different bacteria use different energy sources. Some of them use organic chemicals, like sugars, fats, and proteins. Some of them can actually metabolize inorganic chemicals. There are some bacteria that can actually eat iron. All right, that's an inorganic chemical. And some bacteria are photosynthetic, just like plants are. So those are the bacteria. Now, a close cousin of the bacteria are the archaea. Archaea are also prokaryotic. So the two prokaryotes, archaea and bacteria. Archaea, also prokaryotic. They're also single-celled. But the reason that they're not considered bacteria is mostly because they lack peptidoglycan. They have a cell wall usually, but that cell wall is not made of peptidoglycan. It's made of something else. They tend to live in extreme environments. All right, that's one thing that's characteristic about archaea. They live in extreme environments. And they can include different types. Some are called methanogens because they produce the gas methane. Some are, for example, in the intestines of cows. Cows produce a lot of methane, usually by belching. And a lot of this is due to metabolism by methanogens that's within the cow's guts. There are archaea that are called extreme halophiles. That means that they can tolerate high salt concentrations. So you can find some of them growing in the Great Salt Lake and the Dead Sea, which are very salty environments. And some are extreme thermophiles. They can live at very high temperatures. So, for example, the hot springs at Yellowstone National Park, deep sea thermal vents at the bottom of the ocean that are fed by volcanic activity, very, very hot environments, often approaching boiling point. And, these archaea can actually survive and thrive in those environments. So fungi are next. Fungi are eukaryotes. They have cell walls made of chitin, and that's not always true. There's other materials that certain fungi use for their cell wall. They use organic chemicals for energy. Usually they are decomposers, so they break down decomposing plant or animal matter, and they do that by releasing enzymes into their environment and then absorbing the digested nutrients. There are different types of fungi. There are the multicellular fungi like molds and mushrooms. They consist of masses of mycelia, which are composed of filaments, long filaments called hyphae. So these would be the hyphae, these long stalks that you see. And then we also have yeasts. Yeasts are a type of fungi, but they are unicellular. All right. We also have protozoa. Protozoa are eukaryotes. If you remember learning about protozoa in general biology, they are sort of like a catch-all term. For any single-celled microorganism that is not a fungus, a bacterium, or an archaea. Those are protozoa. So they're a very broad grouping of organisms. They are eukaryotes. They absorb or ingest organic chemicals for energy. A lot of them are modal. They can move, and that can either be through cilia, flagella, or pseudopods, like the example here of this amoeba. This amoeba can actually project its plasma membrane outward and sort of crawl along a surface, and those projections are called pseudopods. They can be free living. They can live on their own. A lot of them you'll find living in the environment, like in pond water or mud. Or some of them are parasitic, where they only derive nutrients by feeding off of a living host. Algae we're probably familiar with, are eukaryotes. They are not plants. A lot of people think algae are plants. They're similar, but they are not plants. They are single-celled microorganisms. Some of them are multicellular, as well. But they have cellulose cell walls, which, if you remember, is the same substance that makes up the plant cell wall. So they are similar in that sense. They are photosynthetic, just like plants are. And like I said before, earlier in the lecture, a lot of the oxygen that's produced in our atmosphere is produced by algae and cyanobacteria, which are similar to algae. So they produce molecular oxygen and organic compounds. Here's one type. This is a pond alga called Volvox. You'll find that one obviously in pond water. Second to last, we have viruses. Viruses are acellular. They are not composed of a cell. All right, they are only composed of very simple materials. They do have genetic information. They have their own genes, either in the form of DNA or sometimes only in the form of RNA. That core of DNA or RNA is surrounded by a protein coat. That protein coat is sometimes enclosed within a lipid envelope. So we have viruses that we call enveloped because they have a lipid envelope on their external surface, and some are non-enveloped. They don't have that. The thing about viruses that you probably learned in general bio is that they are not really considered alive. They can only replicate when they're inside a living host cell, when they are taking over that host cell and using the host cell's machinery like ribosomes. So because of that, because they can't reproduce on their own and they don't have an ATP generating mechanism, they don't have their own ribosomes, those are reasons that we don't consider them truly living. All right. That doesn't really matter when it comes to human diseases because, as we know, viruses, whether they're alive or not, still cause disease. Lastly, in microbiology, we study multicellular animal parasites. So these are eukaryotes. They are multicellular animals. And usually when we're talking about these parasites, we're referring to the worms. So we have flatworms and roundworms, and in general, we call these worms helminths. All right. The reason why we study them in microbiology, as you can imagine, they are macroscopic, they're large, but they are microscopic in their early stages in life. All right. So here's an example here. It's a parasitic guinea worm, which is a disease that's almost completely eradicated on Earth, but you still find it in parts of Africa. This is a subcutaneous worm that causes a very painful infection, and really the easiest way to get rid of it is wait till it sort of pokes through the skin, and then a doctor or anyone really can take a small stick like this and start winding the worm around the stick to physically remove it. It's really thought that the symbol for medicine, which is shown here, is actually a representation of this process. You can see that in that symbol for medicine, you usually see what looks like really two snakes, but might be really a worm being wrapped around a stick. So it's thought that that's where this symbol originates from. So classification is something we're going to discuss more in Chapter 10, but classification in this textbook anyway, and what most scientists agree, is that we classify organisms based on three domains: the bacteria, the archaea, and the eukarya. All right, they're all related. They all originated from a common ancestor. All right, if we look at this, this sort of tree of life, the eukaryotes have several kingdoms, and they're made up of animals, plants, fungi, and protists. Those are all the eukaryotes. We can see them here. So again, we're going to focus on that more in Chapter 10. What I'd like to get to now is a brief history of microbiology. We're going to look at some of the major discoveries that were made leading up to where we find ourselves today. So first of all, the ancestors of bacteria were the first life on Earth. We have to recognize that 3.5 billion years ago, that's when the first life on Earth is thought to have emerged, 3.5 to 4 billion years ago. They were single-celled organisms that were most likely very much like modern-day bacteria. But it wasn't until the year 1673 that humans actually observed the first microbes. So that's where we're going to kind of start, is the first observations. So first of all, in 1665, a guy named Robert Hooke, he was sort of an amateur microscopist. Yes, that's a word. He was a guy that kind of made his own microscope and look at things under that microscope. He reported that living things were composed of little boxes that he called cells. So he was the first to coin that term cell. What he was looking at was cork cells. So the same cork that's used to make stoppers for wine bottles. He would look at that, and of course it's wood, so it's made of cells. And he would see that under the microscope. He saw that in lots of other different living organisms too. This is kind of what started off, this was the first observation that started off the cell theory, that every living thing is made out of cells. So we'll get to the cell theory later though. A few years later, a guy named Anton van Leeuwenhoek described live microorganisms. So Hooke observed cells, but van Leeuwenhoek described microbes. So he had a microscope that was better than Hooke's, so he was able to really see these microorganisms a little bit better. Now, what he called them was animalcules, which is a combination of animal and molecule. So he would look at things like pond water, rainwater, anything he could really get his hands on, and in a lot of those samples he saw little things swimming around, and that's what he called them, animalcules. So that brings us to two competing theories that were going on around this time. Now, if we have to think back a little over 100 years ago, that's not that long, people still believe that some forms of life could emerge spontaneously. That, in other words, life did not have to come from other life, that life could just magically appear. That kind of gets at the heart of the debate between these two theories: spontaneous generation, which was the prevailing theory of that time, that the hypothesis that living organisms arrive from non-living matter and that it is what they called a vital force. That's what forms life. All right, so some sort of vital force that creates life. Biogenesis, on the other hand, was the new emerging theory or hypothesis that living organisms do not arise out of non-living matter, but they have to arise from pre-existing life. All right. Now, what we're going to look at are some of the experiments done during those years to sort of prove one or the other. So the first, it was in 1668. Francisco Redi filled 6 jars with decaying meat. He left 3 jars covered with a fine net, and then 3 jars he left open. Now, the reason he did this was that if it was true that there was really a vital force that formed life, then that vital force should be able to get through the fine net, whatever that vital force was. If it was God or spirits, should still be able to get through the fine net. So, as we can predict, the jars that were covered with a fine net did not have any maggots growing, whereas the 3 open jars had maggots growing. So from where did the maggots come? That's the question. Obviously now we know that it's because flies laid their eggs on the meat in the 3 open jars. They couldn't get to the meat in the jars covered with a net. We know the purpose of the sealed jars, mentioned that. Now, my question is, what does this prove? Does it prove spontaneous generation or biogenesis? All right, so give that a thought for a second. And the answer really is that it proves biogenesis, that life cannot just magically appear. All right, life has to come from other life. So fast forward to 1745. John Needham, another scientist, put boiled nutrient broth into flasks and then sealed the flasks. So he transferred boiled nutrient broth that was sterilized, so this could be chicken broth or corn broth. He sterilized it by boiling it and then transferred it into flasks and then covered them. So in that case, he saw microbial growth. It turns out that microbes did grow in that broth that was sterile. All right. So in his mind, was he proving spontaneous generation or biogenesis? That's the question. All right. So think about that. And he was what he thought he was proving was spontaneous generation because he sterilized this broth, he transferred it to a flask and sealed it, and yet something still grew. However, where did those microbes actually come from? If you think about the experiment. So take that into consideration. He boiled broth to sterilize it and then transferred it into flasks and then covered them. All right, where did the microbes come from? So think about it for a second. The answer really is they could have come from anywhere, most likely the air. So by transferring sterile broth into a flask, you could possibly introduce microorganisms from the air, and that would contaminate it. Or maybe the flasks themselves weren't properly sterilized. We don't exactly know, but likely he did something wrong with his sterile technique. But in his mind, he was proving spontaneous generation. Now, Lazzaro Spallanzani, an Italian scientist, saw the work of John Needham, so this is about 20 years later, and kind of thought the same thing that we thought. Maybe there was some kind of outside contamination. So what he did was he modified the experiment. He put nutrient broth into a flask and then heated the nutrient broth directly in the flask and then sealed it. So there was no transferring of broth between one container to another. And as a result, he saw no microbial growth when he did that. So of course he was proving biogenesis. So why this happened, by the way, is up until this point, nobody knew that microbes were in the air. All right, that hadn't been proven yet. So John Needham didn't realize that by transferring sterile broth from one container to another, he might accidentally contaminate it. So back to the theory of biogenesis by this time, by the late 17 to mid 1800s, was starting to gain a lot of traction. So in 1858, Rudolf Virchow was the first to say that he made a theory that says cells must arise from pre-existing cells. A cell cannot just appear out of thin air. And that is what we call the cell theory. All living things are composed of cells, which kind of ties in John Hooke's work. and all cells have to come from pre-existing cells. So, and by the way, of course, if you want to think about it this way, there obviously is some exception to the cell theory in that the first organisms on Earth, the first cells on Earth, must have come from nothing. But we don't exactly know what the recipe, what the primordial soup was to make that happen. We don't really know at this point. But at one point that must have happened. But ever since then, all cells have to come from other cells. So an important scientist was a guy named Louis Pasteur. He was considered sort of one of the founding fathers of microbiology. Now, he proved in 1861 that microbes are present in the air, which, remember, I said back in John Needham's day, they didn't know that. So Louis Pasteur demonstrated that. He first did an experiment where he put nutrient broth in a flask, heated it, and then left it open to the air. He took nutrient broth in a flask, heated it, and then sealed it. Of course, the one that was left open to the air had microbial growth. The one that was sealed had no microbial growth. So this kind of started to prove in his mind that microbes are actually in the air. That's where this is happening. And he took that whole theory a step further and came up with this kind of ingenious experimental setup. He designed this flask with an S-shaped neck on it. All right, so this design, how it works was he put nutrient broth in this flask with a long glass stem, and then bent the stem using heat into an S shape, and then boiled the broth in there to sterilize it. Now, his theory behind doing this was that the shape of the neck, the S shape, would be such that microbes would not be able to get in and contaminate the flask, contaminate the broth. So a microbe might be able to get in here, but it's highly unlikely that it would make its way all the way up this part of the stem and then down. All right, so this was another way of proving that he could keep this whole apparatus open to the air, but that microbes wouldn't actually be able to get in and grow. So that was, again, a way of proving that microbes are in the air. And as it turns out, he was right. This flask, when sterilized and with a neck shaped like an S, did not grow anything. This is actually a picture of one of Pasteur's original flasks at the Pasteur Institute in France. So this is 150 years old, and there's still nothing growing in it. That's how kind of true this setup was. So that kicked off what's called the golden age of microbiology. A lot of important discoveries in microbiology were made between 1857 and 1914, and to that end still are being made today. The golden age began with Pasteur's work. Discoveries included the relationship between microbes and disease, immunity, and antimicrobial drugs. So a very important time in microbiology. So Pasteur showed another thing. Pasteur, by the way, was a winemaker by trade. He made wine. And what he showed is that microbes are what are responsible for fermentation, which is the conversion of sugar into alcohol in order to make beer and wine. That's fermentation. Nobody really knew how this process worked. Scientists maybe thought air converted the sugar into alcohol, but he proved that it was microorganisms that were doing it. He also figured out that microbes are responsible for the spoilage of food. So, for example, being a winemaker, he noticed, or a lot of winemakers noticed at the time, that sometimes your batch of wine would spoil because occasionally bacteria would get in there that converted the alcohol into acetic acid, which is vinegar. So it would turn the wine and give it like a vinegar taste, and then you'd have to throw it away. So he actually came up with the technique pasteurization, not to preserve milk like we think of it, but it was in order to preserve or kill the bacteria in wine that might spoil it. So he demonstrated that he could do this, that he could kill the spoilage bacteria by a small amount of heat that was not hot enough to evaporate the alcohol, but was hot enough to kill the microbes. And that's really what pasteurization is. It's the application of a high heat for a short period of time in order to prolong the life of food by lowering the number of microbes. So another important theory during the golden age of microbiology was the germ theory of disease. The germ theory of disease is basically that microorganisms are the cause of a lot of human disease. And again, they didn't know this. If someone got tuberculosis, they didn't know that that was caused by the bacterium Mycobacterium tuberculosis. They didn't know that. This was something that was starting to be discovered in the 1800s. So some important discoveries were these. In 1835, Agostino Bassi showed that a silkworm disease, which killed off all the little worms that made all the silk, was actually caused by a fungus. That was a real problem if you were in the silk industry, to have all your worms die. 1865, Pasteur showed that there was a separate silkworm disease caused by another organism, a protozoan. And importantly, in the 1840s, Ignaz Semmelweis, a Hungarian physician, he started advocating hand washing in order to prevent transmission of puerperal fever from one OB patient to another. Now, this disease, puerperal fever, which we don't really hear about nowadays because nobody gets it, it's a disease caused by Streptococcus bacteria that is transmitted during childbirth from mother to mother. So Ignaz Semmelweis noticed that if doctors or midwives who were delivering babies would wash their hands in between deliveries, then the women were less likely to get this disease. This was very important because this kind of started the whole wave of realizing that aseptic practices were going to be very important in medicine. All right. And we'll get to that in a second. Actually, we'll get to it now. This is what I was alluding to. Is in the 1860s, applying Pasteur's work, which showed that microbes in the air can spoil food, can cause disease, a guy named Joseph Lister, who was a surgeon, started using chemical disinfectants to prevent surgical wound infections. And yes, you might realize that Lister sounds like Listerine. Listerine was as a mouthwash that was named sort of in his honor, but no, Joseph Lister did not discover Listerine. What he did do is he used the chemical phenol to sterilize or to disinfect surgical instruments. So back in these days, believe it or not, this is only the mid-1800s, when a doctor would perform surgery, without anesthesia, mind you, they would take their blades or their knives, and after performing surgery on one patient, might just wipe them off with a towel, wipe all the blood off, and then go perform surgery on another patient. Obviously, we now know that that's horrible aseptic technique, but nobody knew this at the time. So it was Joseph Lister that first advocated disinfecting instruments. So in 1876, a guy named Robert Koch proved that a bacterium causes anthrax. So again, we're starting to realize during this time that many common diseases like anthrax are actually caused not by spirits or punishment from a higher power, but they're actually caused by microorganisms. So Robert Koch proved that Bacillus anthracis is the bacteria that causes anthrax. But most importantly, he came up with a series of experimental steps that are called Koch's postulates. And these are experimental steps to guide scientists that to show that a specific microbe causes a specific disease. And we're going to get to Koch's postulates later on in the semester. Now, another important discovery was the idea of vaccination. And vaccination actually has a very old history. So the idea of vaccination originated in India, I believe, around about 1,000 years ago. But it didn't really gain traction in Western societies until kind of more recently. So starting in 1796, Edward Jenner inoculated a person with the cowpox virus, and then that person was protected from smallpox. So Edward Jenner noticed something. In those days, the women who milked cows were called milkmaids, and oftentimes they would contract from the cows, they would contract a disease called cowpox, which is very similar to human smallpox. So this is a disease, cowpox, that can actually be passed on from cows to humans. They would get a mild case of this cowpox disease, but then what he noticed is that would actually protect them from human smallpox. They would never get human smallpox as a result. So based on this observation, Jenner then decided to perform an experiment where he got a volunteer, it was like an 8-year-old boy, I believe, and got this, and it basically took material from cowpox scabs, and injected it into the little boy. And as it turns out, that boy was protected from human smallpox. He never developed it. That was kind of an early experiment, but the concept is there. It wasn't until Pasteur came along when he determined, he kind of figured out how vaccination works. But that's where we get that term vaccination. It's derived from vaxa or vaca for cow. That's a Latin word. And this protection is called immunity. So the whole idea of vaccination was kind of born out of this era. During this time, we also see the birth of modern chemotherapy. Okay, so prior to, around the turn of the century, 1900 or so, we had no drugs to treat infectious diseases whatsoever. All right, so that's really what people started to discover. All right, now for some terms, chemotherapy is a term that means treatment of a disease with chemicals. So it doesn't necessarily mean cancer treatment, but chemotherapy means taking a drug to treat a disease. Now, the chemotherapeutic agents that we use to treat infectious diseases, they can be synthetic drugs or antibiotics. Antibiotics are a special type of antimicrobial drug. They are chemicals that are produced by bacteria and fungi that kill or inhibit other microbes. So antibiotics, when you use that term, they're actually naturally occurring chemicals that are produced by bacteria and fungi. All right. So let's talk about the first antimicrobial drugs. Now, one exception to this is that quinine from a type of tree bark was actually long used to treat malaria very successfully. But other than that, there really were no drugs to treat other infectious diseases. It was a guy named Paul Ehrlich that speculated about a magic bullet, that's what he called it, that could destroy a pathogen without harming the host. So he thought, what if we can come up with some chemical that is toxic to the microbe but not toxic to the host? And in 1910, Ehrlich did that. He developed a synthetic arsenic-based drug called salvarsan, and it was successfully used to treat syphilis. All right, syphilis was a very rampant disease during the time, sexually transmitted, and if it's not treated, can sometimes lead to severe brain damage. So he came up with this drug, and that was really the first one. It was a synthetic drug. It was arsenic-based, which turns out to be obviously somewhat toxic to humans, but it was more toxic to the bacteria. In the 1930s, sulfonamides were synthesized, but probably the most famous antibiotic discovery was the discovery of penicillin. This was a fortunate accident. It was 1928. A microbiologist named Alexander Fleming discovered it. So Fleming was studying Staphylococcus aureus bacteria. So let's look at this plate right here. He was growing, what you see here, these are colonies of Staph aureus bacteria. In one of his petri dishes, he noticed that there was a contaminant. There was a colony, some organism growing on there that was clearly not Staph aureus. The colony is way too big, doesn't look the same. So that's normal. That happens once in a while. You get contamination. But luckily, he took a second to observe it. Now, what he saw was that around the colony, there was an area of inhibition of bacterial growth. So you can see that the colonies of staph in this area around this big colony, they're very small. So he thought, wait a minute, maybe this penicillium, well, he didn't know that's what it was called, but he said maybe this contaminating colony is secreting a substance into the agar, and it's killing or inhibiting the bacteria around it. And as it turns out, he was right. All right, that fungus that was growing there turned out to be penicillium. It was named penicillium, and the antibiotic that it produced is penicillin. And a few years later in the 1940s, penicillin was tested clinically, it was mass-produced, determined to be safe, and it was really thought of as a miracle drug, and it was sort of given out like candy because we didn't know about antibiotic resistance back then. So, what we'll look at lastly, or next, is some modern developments in microbiology. Now microbiology has a lot of different subspecialties. So if you study bacteria, you're actually a bacteriologist. That's bacteriology is the study of bacteria. Mycology is the study of fungi. Parasitology is the study of protozoa and parasitic worms. And immunology is the study of immunity in the immune system. And this is, of course, is still a very active area of research. Vaccines and interferons are being investigated to prevent and cure viral diseases. And a major advance in immunology happened in 1933. So a scientist named Rebecca Lansfield was figured out how to classify streptococci, strep bacteria, based on their cell wall components using antibodies. All right. Now, for any of you guys who have heard of the term like Group A strep or Group B strep, some of you may have heard that or not. The kind of the kind of strep bacteria that you get when you have strep throat is called Group A strep. Now, this was all because of Rebecca Lansfield. She came up with those groupings based on their cell wall components and how they reacted with antibodies. Another thing I always like to point out here is that was a big deal for a woman to be a scientist in 1933. So that was a very big discovery, very important discovery. Virology is the study of viruses. It was Dmitry Ivanovsky and Wendell in 1892 and Wendell Stanley in 1935 that discovered the cause of the mosaic disease of tobacco as a virus. So these two scientists discovered that a certain disease in tobacco plants was caused by a virus that they called tobacco mosaic virus. Why is that important? That was the first virus ever discovered. So it really wasn't until the mid-1930s that we even knew that there was another class of microorganisms called viruses that exist. And a lot of that has to do with the fact that viruses are much smaller than bacteria, so they're harder to see and they're harder to grow. And it's really electron microscopes that have made the study of viruses really possible. So back then, in 1935, the electron microscopes were brand new, and you could start to see some of these ultrafine things and structures. So another important area of study that's kind of related to microbiology is recombinant DNA technology. You learned about this in general bio toward the end of the semester. So first we'll start with some terms, what we mean by this. Microbial genetics, the study of how microbes inherit traits. Molecular biology is the study of how DNA directs protein synthesis. Genomics, the study of an organism's genes, or the study of an organism's entire genome. That's genomics. Now, what do we mean when we say recombinant DNA? Again, you learned about this in general bio. It's when you take DNA from two different sources. And this was brand new in the 1960s. Paul Berg was the first to do this. He took animal DNA and inserted it into bacterial DNA. And then, as it turns out, the bacteria then produced that animal protein that was coded for by the animal DNA. That's what recombinant DNA technology, or sometimes it's called genetic engineering, is. It's taking DNA from two different sources. Now, this obviously all has to do with microbiology because most recombinant DNA studies are done in bacteria or other microorganisms. So, some history behind recombinant DNA technology and molecular biology and all this. It's a relatively new area of study. In 1941, Beadle and Tatum showed that genes are what code for a cell's enzymes. So during that time, there's a big debate. What actually is the genetic information? What is the material that's passed on from cell to cell? And what provides the instructions to make things like enzymes? And they showed it was genes. In 1944, a similar discovery by Avery, McLeod, and McCarty showed that DNA is the hereditary material. It's also what's passed between parents and offspring. Watson and Crick proposed the model of DNA structure. They're the ones that came up with that whole double helix shape of DNA. No one really knew what the structure or what the shape of DNA was at the time. And Jacob and Monod discovered the role of messenger RNA in protein synthesis. All right, so put that all together, and then you have basically the foundation of what we know about genes and molecular biology. And of course, there's more to the story than that. Here are some selected Nobel Prizes that have been awarded the past 100 or so years for microbiology research. The most recent one was by the French team that discovered the causative agent of AIDS, the HIV virus. Believe it or not, that wasn't awarded until 2008, and they discovered that in the 80s. So sometimes Nobel prizes take a while. The same year, Harold Zurhausen was given the award for the role of HPV in causing cancer. So you can go back, and these are just sort of, I put them in there because they're kind of interesting, to show, some of these important discoveries and when they were made. So next we're going to talk about microbial ecology, how microbes play a role in the environment. So we mentioned this before, that bacteria are the ones that convert carbon, oxygen, nitrogen, sulfur, and phosphorus into forms used by plants and animals. They're very important in recycling all these nutrients in the environment. Bacteria, microbes, also do a good job of helping to detoxify pollutants and organic matter in sewage. And we call this bioremediation, when microbes are used to sort of clean up waste. So we know that they play a big role in degrading sewage. If you go to any sewage treatment plant, they have these big holding tanks containing all the human waste, and part of that is to allow bacteria that they add to break that down into smaller components. They can also detoxify pollutants like oil and mercury. So what this is showing is a couple of workers spraying bacterial cultures onto rocks that have been where there was an oil spill. So bacteria can be used to even break down oil. And speaking of that, alkanovorax. These are a species of naturally occurring oil degraders. They live in the ocean and other aquatic environments, and they naturally eat oil, if you want to think of it that way. So this is why, for example, when there was that whole Gulf of Mexico oil spill, that it actually seemed to resolve itself a lot faster than most people thought. And part of that was due to these naturally occurring bacteria that can help to degrade oil. So if you look at them under a microscope, you see that they're forming this sort of bubble around this area. This area here that's labeled alkane phase, that's really a fancy chemistry term that means this is an oil droplet right here. And they're surrounding it, they're releasing enzymes to break down those oil components, those hydrocarbons, and they're absorbing the products of the breakdown and using it for energy. So another important role that microbes can play are as insecticides. There are some bacteria, some microbes that are pathogenic to insects, and it's thought that these can be an important alternative to chemical pesticides, especially in agriculture. Because in agriculture, if you're working on if you own a massive, 500-acre farm, and all of your crops die because of a disease, that's a big deal. So rather than spray chemical pesticides, it's possible to use biological insecticides, bacteria whose infections are toxic to insects. And the best known example of this is a bacterium called Bacillus thuringiensis. Infections by this bacteria are fatal in insects, but they're harmless to other animals, humans, and to plants. So here is a Bacillus thuringiensis cell. The cell is actually collapsed, but you can see that what it releases are these little protein crystals. These crystals, when they're ingested by insects, they get lodged in the digestive tract of the insects, and it's fatal to them. Now, those toxic little crystals don't get lodged in our digestive tract because our digestive tract is significantly larger than that of insects. So these infections are very fatal to insects. And in fact, what they've done is they've taken the gene for this toxic crystal out of Bacillus, and they've inserted it into the genome of many different of corn. So now the corn itself is able to actually produce the toxic crystals to prevent being eaten by insects. All right, so some of you may have heard of this, that genetically modified corn in the United States, for the most part, is also called BT corn. All right, if you've ever watched any documentaries on, food production in the United States, you've probably heard this. But BT corn is called that because it has one of the genes from BT, Bacillus thuringiensis. It has the gene to make this toxic crystal that kills insects. So biotechnology is kind of what we're getting at. That whole idea of modifying organisms or using organisms, microorganisms, to do things that we want is really what biotechnology means. All right. Now, recombinant DNA technology is a big aspect of this. Anytime in biotech, when you need to force a microbe to make something that you want, you use recombinant DNA to do that. All right. This is a technique that has enabled bacteria and fungi to produce a variety of proteins like vaccines and enzymes. A couple examples are insulin. We mentioned that earlier. Insulin is now produced by genetically modified E. coli. Back in the day, prior to 1978, Insulin, if you had diabetes, insulin was obtained usually from pigs or cows. So they would use pig or cow insulin, which is not as effective as human insulin, and sometimes it caused an allergic reaction in people with diabetes. So they actually can now make human insulin ever since 1978, and they did that by taking the gene for human insulin and sticking it into E. coli, and then E. coli in turn made the insulin protein. So I want to make a couple statements about our normal microbiota, of course. The normal microbiota are all of the quote-unquote good bacteria that live on or inside us. And it's not just bacteria, by the way. We're colonized mostly by bacteria, but also by yeasts and certain other microbes, but sometimes I'll slip up and just say bacteria. So that's what we call them, our normal microbiota. Microbes normally present in or on the human body. Some of them are actually very good. They do good things for us, which we'll get to. Now, sometimes you might hear the term normal flora. That term has been replaced by microbiota. Flora is a kind of an antiquated term when bacteria used to be classified as plants. So we say the term normal microbiota. Now, what are the good things that they do for us? They can prevent the growth of pathogens. So basically, if you look at every single space on the human body, like on the surface of the human body or in all of the orifices of the human body, like our whole digestive tract, pretty much every space that is possible to be colonized by microbes is colonized by microbes. And so because of that, those normal microbiota compete with pathogens for space and nutrients. So it makes it less likely that a pathogen will take hold if you have all these other competing microorganisms. Normal microbiota produce some important growth factors for us, like folic acid, which is a type of B vitamin, and vitamin K. So in particular, microbes in our large intestine, some of them produce folic acid and vitamin K, and we absorb it. So that's good for us. Now, besides some of the good things that they do, we're kind of going to, now we're going to transition into some of the bad things that microorganisms can do. We're going to transition more into disease. So resistance, the term resistance, is the ability of the body to ward off disease. And some of the factors that allow us to resist disease include our skin, as long as it's intact, our stomach acid, and various antimicrobial chemicals that we make. So before we talk about some pathogens, we have to mention biofilms. Biofilms are microbes that tend to grow in large colonies. They can attach to solid surfaces and grow into masses. They can grow on rocks, pipes, your teeth, and medical implants. Now, why do we care about biofilms? They are playing a more and more of an important role in causing infection, and they're often resistant to antibiotics. So not all bacteria can form biofilms, by the way, but some can, and when they do so, they grow in these large communities, and they all release slimy substances into their external environment. And that slimy substance actually allows them all to stick, and it provides them some kind of shelter from things like chemical disinfectants. So you'll find biofilms growing everywhere. Even if you're at a pond or a lake, and you notice sometimes that there'll be a rock in a pond or a lake that's kind of slimy, that's usually a biofilm of microorganisms. When your teeth get all sort of, when you haven't brushed your teeth in a little while, and then they get that film on them, that's a biofilm of bacteria. In that case, the bacteria are Streptococcus mutans, and they're the ones that can cause decay. But importantly, they can grow on medical implants, and this is a problem because when bacteria grow in biofilms, like I said, they're more resistant to chemical disinfectants, often very much so. So if they're growing on a medical implant, which is going to be put into the human body, that's a guaranteed infection. So with that said, we're going to focus now on some infectious diseases. Really, we're going to look now at emerging infectious diseases. These are diseases that are either new or increasing in incidence, and probably you've heard of most of these because they're the ones that we're kind of watching out for. The first is Middle East Respiratory Syndrome, or MERS. It's caused by the Middle East Respiratory Syndrome coronavirus, or MERS-CoV. It's actually very related. It's similar type of virus to SARS, which is Severe Acute Respiratory Syndrome, We were hearing a lot about that several years ago. So it's a similar virus. There have been 100 deaths in the Middle East from 2012 to 2014. It probably arose from bats and was transmitted to humans. They also think that it could be transmitted to humans via sick camels. And in the Middle East, camels are more common. So that's that particular disease. So it's a severe respiratory disease, by the way, and has, fairly high mortality rate compared to other respiratory diseases. Influenza A, avian influenza A, I should say, bird flu. There are many, many different strains of influenza. One of them, though, is we sometimes call the bird flu. It's known as the influenza A H5N2 virus. It's primarily a disease of birds, waterfowl, and poultry. But in some cases, it can be transmitted to humans from these birds. So in people working on, poultry farms who work around birds a lot, these are the types of people that have gotten avian influenza. And it's actually a very aggressive flu. And much more deadly than the normal seasonal flu that we see. The thing is, we haven't seen sustained human-to-human transmission yet. So the virus has not really changed in a way that allows it to pass from human to human. It can only go bird to human as of right now. But we're watching out for it, because if it does mutate in a way that it transmits human to human, we could have a very serious outbreak. Probably a lot of us have heard of MRSA, especially if you work in the hospital. MRSA stands for methicillin-resistant Staphylococcus aureus. We mentioned Staphylococcus aureus earlier. It's a common staph bacterium. And a lot of us, like 40% of us, have Staph aureus living on our skin. It's a common human microbiota. But there's a specific strain of it that's highly drug resistant. That's what MRSA is, methicillin-resistant Staph aureus. And it's not just resistant to the antibiotic methicillin, it's resistant to a whole list of antibiotics. And this happened over the last few decades. In the 1950s, there was penicillin resistance was reported in Staph aureus, because, of course, penicillin only came out in the 1940s, and people were likely overusing it, misusing it. In the 1980s, Staph aureus developed resistance to methicillin. In the 1990s, MRSA resistance to vancomycin was reported. So in the 90s, some strains of Staph aureus that we call MRSA were resistant to every antibiotic except vancomycin. There were really no other useful antibiotics to treat it. And there was really a fear that we would that we would sort of blow through vancomycin as well, that it would develop resistance to vancomycin. And it did in the 90s. We do now have strains that are vancomycin-resistant Staph aureus. So the problem is we're always having to develop new antibiotics. Otherwise, bacteria will always develop resistance. That's just what they do. So antibiotic resistance is something that happens naturally as we continue to use antibiotics. So another emerging infectious disease is West Nile encephalitis, and this has been around for a while now. We first heard about it in the United States really in 1999. It appeared in New York City. It was first diagnosed in 1937 in the West Nile region of Uganda, which is where it gets its name. It's transmitted, or it's found in non-migratory birds in 48 states, and it's transmitted between birds and to horses and humans by mosquitoes. So we all know about West Nile because it's been a problem in Massachusetts for quite a few years now. You always hear about a few cases, and it causes encephalitis, which is, a swelling of the brain. Now, in most people, it's not a life-threatening illness. Most people recover, in other words, but in some people, that encephalitis can be very severe and it can be deadly. Another disease is bovine spongiform encephalopathy, also known as mad cow disease. Now, mad cow disease is caused by a prion, which is not a microorganism that we've talked about so far. A prion is actually just an infectious protein. It is not living, it does not have DNA, but in fact it's just a protein that happens to be infectious. And we're going to learn more about that in chapter 13. But basically, when you you get this disease from eating contaminated meat. So the prion is only found in the central nervous system of infected cows. So let's say the meat was tainted somehow because during the slaughtering process, some of the cerebral spinal fluid from the cow accidentally made its way onto the meat. Now, if you ingest that meat, this prion, by the way, is very resistant to heat. So heating will not kill it like it kills other microorganisms. It would enter your bloodstream, eventually make its way into your brain, and then it would cause destruction of brain tissue, all right, where you get these holes in the brain tissue. And that's where that term spongiform comes from, is that your brain tissue starts to look spongy from this dead area. So prions also cause other diseases that are similar, like Creutzfeldt-Jakob disease is an inherited genetic disease that's similar to mad cow. It pretty much works the same way, but instead of being acquired, it's inherited. So when you acquire mad cow disease, what they actually call that is variant CJD, variant Creutzfeldt-Jakob disease, when it's acquired in humans. So part of the problem of why cows were getting mad cow disease is they may have actually gotten it from sheep originally. So what they were doing was when sheep would die, presumably they would die from this disease, they would take the sheep and actually use them to make feed to feed the cattle, including parts like the brain. Now, of course, to do that, they would heat up the sheep and render the meat. That's what they call it when they basically boil, animal parts. They render it. But boiling doesn't kill the prion. So then these cows were being fed prions from sheep. They were getting the disease, and then the disease was being passed to humans. So we've all heard of E. coli. All right, we mentioned it earlier, Escherichia coli. Now, I want to be clear, E. coli gets a bad rap, and I kind of feel bad for E. coli. I guess I shouldn't. It doesn't have any emotions. But E. coli, most strains of E. coli are perfectly harmless. We all have E. coli living inside of our large intestines. It's part of our normal microbiota, and it does nothing harmful to us. It produces good things for us. It's our normal microbiota. However, there are a few toxin-producing strains of E. coli that do cause human disease. And the best-known example is E. coli 0157H7, which is a form of a form of E. coli that produces a toxin that causes diarrhea. So that toxin, when you ingest the E. coli, it releases the toxin. The toxin basically kills the cells that line your small intestine and large intestine, and then that leads to water rapidly flowing into the large and small intestine, leading to diarrhea, sometimes bloody diarrhea. So it was first seen in 1982. This is a relatively new strain. Now, Ebola, this is one that probably up until the 2014 outbreak, not many people probably most of us had heard of it, but maybe didn't know much about it. This most recent outbreak was the first major one in a while. It was, in Guinea and other parts of Western Africa. They say hundreds killed. I'm assuming, now that I'm looking at this, that this textbook was maybe just published in the middle of the outbreak, because I know at this point, I think there were like 10,000 people that had died from Ebola in the most recent outbreak. But anyway, Ebola hemorrhagic fever is the name of the disease, and it's caused by the Ebola virus. It causes fever, hemorrhaging, and blood clotting. So basically, this virus attacks the lining of your blood vessels, and that can cause blood to leak out. That's hemorrhaging, and it can cause blood clotting. It's basically a horrible way to die, and it's very fatal. Some strains are up to 90% fatal, and there's really not much that can be done. There's no vaccine right now. There's really just supportive care, rehydrating people, keeping them as comfortable as possible. It's transmitted via contact with infected blood or bodily fluids. All right, so luckily, even though it's a very fatal disease, it does require fairly direct contact. It's not transmitted, through the air very easily like some other viruses are, but it's very deadly. It comes from, the prevailing theory is that it comes from bats, and it's transmitted to humans periodically, and we see these outbreaks. So, the next one, cryptosporidiosis. All right, this one not a lot of people have heard of. It's a very important diarrheal illness, though. So, and the reason why we haven't heard of it is because in the United States we have very clean drinking water, and we don't have a lot of diarrheal illnesses. So, like cryptosporidiosis, E. coli 0157, other strains of E. coli that cause disease. These are all diarrheal illnesses, and a lot of times they're transmitted because of contaminated drinking water. But in the United States, like I said, we don't have as much of a problem because our water is fairly clean. But this disease is caused by a protozoan, Cryptosporidium. It was first reported in 1976. And in developing countries, it causes 30% of diarrheal illness. So it's a major cause of diarrheal illnesses. It doesn't sound so bad when you say the word diarrheal illness, like you're not going to die from it, but you have to keep in mind if you're living in a third world country without clean drinking water and you have horrible diarrhea, you're losing a lot of fluid, and then you're trying to replace that fluid with more contaminated water. And a lot of times these climates are very hot, and so people can die from things like dehydration and loss of electrolytes due to diarrheal diseases. Lastly is acquired immunodeficiency syndrome, AIDS. We've all heard of AIDS. AIDS is the name of the disease that's caused by the human immunodeficiency virus. It was first identified in 1981, and now we know it as a worldwide epidemic that is infecting 33 million people total worldwide, with 7,500 new infections every day. And that's actually up a little bit higher than it used to be. I think it was around 6,500 before. So it's primarily sexually transmitted. It affects males and females. And if we look at the statistics, at least these are from 2009, so these might be a little different now. But in the United States, 26% of people with HIV or AIDS are female and 49% are African American. So just like, unfortunately, many diseases tend to disproportionately infect certain populations, that's what we're seeing is the African American communities, disproportionately affected by AIDS. So we know this disease. We're going to spend a lot of time talking about HIV and AIDS in a later chapter. So what I like to do is a lot of times I'll end a lecture with a slide with study questions or other kind of quick activities just to kind of reinforce what we've learned. So you can take this time to pause and do these to reinforce what you've learned. Classify the following types of microbes as either a prokaryote or eukaryote. So we have bacteria, archaea, algae, fungi, protists, multicellular animal parasites. List the major discovery of each of these scientists. That's just a few of the ones that we talked about. And which of these on this list is a correct scientific name, which is correctly listed. All right, so take that time and do your homework, and we are done chapter 1.